Hi everyone, I'm Pranika Singh and I'm working at Edelweiss Connect at Basel, Switzerland under the supervision of Thomas Exner. Today I would be talking about the role of data management and quality control and why is it important for an efficient data analysis to get and answer your scientific questions. My talk is divided in three parts. The first being data management, why it is important and how we can implement it. Second being quality control, why it is important and how it should be done. Third, I will give an example of a case study I worked upon, analyzed the data, and found some very interesting results. So the data management is based on fair data principles. These are the set of guiding rules which help you make data findable, accessible, and reusable both by humans and by machines. How do we do that? We have metadata, so we can assign that with globally unique and persisting identifiers, which can then help us retrieve it using a standardized protocol. Also, it's very important that we use a very formal, accessible, shareable, and understandable knowledge to define this metadata so that it could be shared and used by the community who wants to use this data. Also, it's very important that the attributes which we use to identify and define this data should be accurate and relevant so that they can distinguish one observation from another. In the N3 project, I implemented data management with the help of the fellow ESRs. We formulated different ways to make this identifiers to distinguish one sample from another, one experiment from another. So we set these rules before even we conducted the experiments to create the sample IDs and experimental IDs so that we can help us distinguish one sample for another. In addition to that, we also formulated some instruction manual for filling in these metadata files, which also helps in defining and also distinguishing one sample to another. For example, what is the target cell type? What kind of iPSC cell lines we use. So we created a unique way of formulating iPSC cell IDs, plate format which was used, exposure scenario. So all the different attributes which would help define the sample and which are important with respect to the experiment to be known before you do any kind of analysis. And once we have this metadata and the raw data, I uploaded it to the Edelweiss data, which is a knowledge base which was used, which could then be used and for accessing and for analyzing the data downstream, which helps in visualization and interpretation. So after you have your data, after the data was integrated and uploaded and loaded on the Edelweiss data, I also generated an automatic script which could help you select the data you want to analyze and you want to have your scientific questions answered by. So you have an option of selecting the experiment or the case study you want to work upon. You also have an option of selecting multiple experiments. And after you have those experiments selected, you will get an option of selecting the different metadata fields and variables which you have in the metadata files and choose what you are interested in. Once you have your data selected based on what you are interested in, there is a downstream workflow for analyzing this data now for differential expression analysis. Before that, you have also an option of quality control, and then you have option of normalization and differential expression analysis using the famous package called DESEC2. With this, you have also additional options of creating heat maps and doing your downstream analysis after you have the set of differentially expressed genes. This section, I would like to bring your attention to why quality control is important before you conduct any type of downstream analysis like differential expression analysis. So quality control is the most important and the foremost step which should be done before data analysis. And why do I say so? Because it will highlight the most important issues or the outliers or noise if they are present in your data. So that before you do the downstream analysis and get your results, you see if there are some issues in your data which you have to address before you start doing the analysis and get your results. And thereby it will help you identify the most significant and the most important and the most identifiable data, which would then help you answer your biological questions. I would like to quote some examples which I came across where I used quality control to detect some issues. As you can see in this PC on the left-hand side, these set of controls, so all these red points actually represent the samples which belong to a group of controls. They are biological replicates. So ideally, they should be grouping all very together like all of them. But you could see that all these controls which are on the left-hand side are quite dispersed as compared to the controls which are on the right-hand side, which have the PC1, the variance of 30%. And when I went back to the raw count, I saw that there are some genes which contribute to this variance. So the genes have different read counts starting from this control to this control. And this is what you see in the PCA. So there are some set of genes which contribute to the 30% of variance which you see in different control groups. So you could also filter, you also have an option in the script to filter these kind of genes which show such variable or such low read counts that would actually influence your downstream analysis. Another example which I want to quote is about the box plot. As you can see in the box plot that there are these treatments which are done at 24 hours and the concentration was 5 μm. As you can see that the first two replicate looks very similar to each other, but the third one has absolutely very, very low read counts. And you could also see the issue with another one in which you have 2.5 μm and 24 hours. You see that for the first two replicates, they're very similar, but the third one not. And when I went back to the raw distribution of data, I saw that this third replicate here has very, very low read counts as compared to the first two replicates for the genes. And this contributes to what you see here. And the same was for the other concentration, which I saw here. That's why it is important that you remove such kind of samples which show such variance before you do the analysis. Otherwise, your analysis would be based on this, in which you have so much variability in the replicates, which should be considered before you do the downstream analysis, as this will affect your downstream results. Diving more deep into it, I also try to look at the Pearson correlation coefficients, which were calculated by my fellow colleague, Zara Mazidi. After looking at the correlation coefficient for this particular box plot, which I saw that their correlation coefficient were also very, very low. So for the first replicate, it was for the first replicate and the second replicate here, you could see the third replicate show only 0.09 and 0.09, like 9% similarity. So that actually gives you an alarm that there is something wrong here and you have to remove the samples before you consider them for analysis. And the same was seen for 2.5 μm2, that the third one was 0.10 and then 0.10 similar to the other two replicates. So only 10% similarity. So before you do the analysis, it's important that you look at different parameters and then select your samples for doing any downstream analysis. So before I did the analysis, I removed the samples as they were not so similar to the group which they belong to, and then conducted the analysis. There's another example which I would like to give you. This is another set of samples which were analyzed, and we saw that for these three groups, we saw that it is only 0.79. So when the analysis was done by using all these samples, it was seen that we saw some differentially expressed genes at a lowest concentration, but not at the higher concentration, which was 50 μm. But after we removed this outlier from the data, we saw that it improved the quality of the data. Now you see no genes at 5 μm, but genes at 50, and no genes at 50, but only genes at the highest concentration, which made sense and was validated by other literature studies as well. So it's very important that you look at your data as raw data before you do any kind of normalization or analysis like differential expression analysis, so you could detect such kind of outlier and noise in your data. Now I would be talking about the cadmium chloride case study, for which the experiment was done by Vidya under the supervision of Paul in Amsterdam. In this, I wanted to look at how if iPSC cells could help us conduct these metal exposures, study what is the utility of them, and can we resolve the temporal alteration of the pathways which are induced by cadmium, like metal response pathway and oxidative stress pathway. The compound which was used was cadmium chloride, and there were 10 sets of time points which were selected for measuring the temporal expression of the genes, and the concentration was taken as 5 μm. In this slide, you could see that these are the set of differentially expressed genes which were found in all these time points. And then I looked through how they're expressing in different time points. And there are two sets of clusters which are formed by the gene. One is the one which you see in the lower end, that they are the genes which start upregulating even in the first hour, and then they keep upregulating even until the end. But then there are some genes which you see are upregulating, then going to downregulation, or downregulating and then going to the upregulation. So there were a set of 2 or 3 clusters which were found in these pathway, in these set of differentially expressed genes. Then there are, in the lower end, you see, you might know about the metallothioneins, which contribute to the metal response pathways. And we saw that these are highly expressed, highly upregulated genes, which were found in these data sets. And that's why through various literature studies, I know that the metal response pathway is triggered by cadmium. I looked very close to it to find what is the temporal relationship of the genes with the pathway. And in this figure, you could see the x-axis represent that how much percentage of the expression value is retained after 24 hours. So going from -20 to 120. And these represent the timeline or the time point at which the first maxima was observed for the genes. You can see all the metallothioneins here give the first maxima or the highest expression at a very early hour near 4 and 6, between 4 and 6. And most of them have their 60% of expression retained even at 24 hours. So this shows that the metallothionein or the metal response pathway stays active throughout the timeline for which we have collected the data, for which we have conducted the experiments. And they start expressing in a very early hours, and they maintain the expressions even throughout the timeline. They go a little bit down, but even when they go a little bit down, they maintain the expression up to 60%, which is very high and indicate that this pathway was highly activated throughout the timeline. These are the different metallothionein gene profiles, which I try to see and view if they make sense, if they are showing the same expression pattern. And through this, and if you take an overview of this, you see that yes, they show very similar pattern. So I started plotting the normalized counts to see how they are expressing, and you see that even MT1E, MT1F, MT2A, and SLC30A1, they all show very similar profiles even in their normalized counts. Then I also looked at their fold changes, how the pattern looks like in fold changes. And you see that even in the fold changes, the expression or the gene profile gives a very similar view. And with that, I also saw how the expressions look like, the profiles look like from 0 to 24 hours. The earlier ones were from 10 to 168 hours. And you could see even for this timeline, which is more expanded from 0 to 24 hours, we saw that, yes, they show very similar expression patterns. So it's not only that because we have 0 to 24 and then 72 to 168. It's not only what we see here. We also see it in the time points between 0 to 24 hours that, yes, they show very similar gene expression profiles. And you see the similar thing in the fold changes as well for the first 24 hours. So this indicated that all these metallothioneins belong to the cluster, which you see in the end of the heat map here. And also that the temporal expression also contribute to the activation of the metal response pathway, which remain activated throughout the timeline. We did similar thing for NRF2 pathway, and we saw that most of the genes are also activated in the early hours, but most of them go to almost the base level, or their expressions start dropping down in the later hours. So I would like to thank Thomas, Tatiana, and the rest of the Edelweiss Connect team to help me conduct this work, and specifically thanks to Vidya, Cormac, Carolina, Zara, and rest of the In3 team, with whom I have been working closely, and to the In3 project, which gave me an opportunity to work upon this. Thank you so much. And if you have any questions, you can mail me or message me, and I would be very happy to answer.